speaker, but also an ice cream social. So we'll extend the time. So hopefully you'll come and some ice cream. That's just sort of kick off to the summer. Um, but I'm so pleased to introduce Dr. Brad Borlee. He's an associate professor of microbiology, immunology, pathology, so my department here at CSU. His lab studies how bacteria sense and adapt to changing environments, with emphasis on biofilm physiology, microbes in the aerobiome, and bacterial pathogens that affect human and animal health. The Borlee lab trains scientists to connect microbial mechanisms with ecosystem processes and public health using One Health approach that translate foundational microbial physiology into practical tools. And Brad is one of the co-PIs on our NSF-funded Broaden Center that sort of tries to understand how pathogens move in the air. So welcome, Brad.
All right. Well, thanks for giving me the opportunity to talk today. Today, I'm going to be talking about how we are building these One Health discovery networks. And I'm going to put this talk in the context of going from molecules lost the pointer clicker. All right, now we got it. Okay. I'm going to be putting this talk in the context of how we go from molecules to genes, or genes to molecules, to then how these organisms are interacting with each other to colonize different environments, whether it's, a stream or a plant or a human host or an animal, and how they survive and persist in those environments, to lab-reared ecosystems, and put it in the context of what's going on in the environment, how that's affecting human health, and why this is important in a changing world. And so a lot of the talk today is going to be on these sapronotic disease agents. And so these are organisms that you find in the environment. They're very versatile to changing environmental conditions. And that sets them up to be able to adapt to infect to animals and people. And their ability to sense and respond to these changing environmental conditions allows them to better survive in those environments and transition between those environments. And so, kind of preaching to the choir here today, but I'm a big proponent and a fan of employing these One Health approaches to basically make kind of the discoveries that we need to counter the emerging threats that we have to human health and to animals and to our environment. And so what I'm going to try to do is put this in the context of how these interactions between people and these organisms in our shared environment is constantly changing, and how that's important to understand to mitigate the risks of some of these organisms that are in our shared environment. I put it in quotes because we don't share our environment very well, and we shouldn't think of it as ours. But what I'm hoping to achieve today is to really explain how collaboration through multiple disciplines is important to counter these emerging threats to improve human health and animal health. And so one of the things that I'll focus on is how we can basically look at what is going on in the environment to predict kind of how sapronotic disease agents can disperse from that environment to be acquired in an infection in animals and people. But it's also important to keep this in the context of, how can we use these collaborations across these multiple disciplines to fortify global health security, how to improve food and safety and food security. And what's emerging these days is kind of, we have antimicrobial resistance on the rise. How can we use One Health approaches to reduce antimicrobial resistance in populations of bacteria? And so my laboratory has a lot of diverse projects that we work on. A lot of them are integrated into One Health discovery networks. And so today I'm going to be talking a lot about pathogen surveillance and microbial air sampling. And this is funded by the USDA and NSF. And I'll tell you what Broaden is and how Broaden came to be and why this group of people can work together to solve some of these problems that we're encountering in our environment by understanding what's going on with the microbes that are aerosolized into there or emitted into the air. We also do some work on developing vaccines and antibody therapies. I won't be talking about that today, but those are directed at these sapronitic disease agents that are surviving in the environment as biofilms. We also have some activity in this area of looking at what are the bacteria in the guts of insects, specifically mosquitoes and midges. And now we're moving into the New World screwworm, thanks to some pilot funding by the One Health Institute. But in that context, and I won't have opportunity to talk about that today, but we're looking at how we can understand the microbes that are associated with these insects, how we can manipulate that microbiome to alter the capacity of those insects to basically survive in the environment and transmit infectious agents. And so what I want you all to do, I'm going to put this in the context of human health. And so I want you to put on your white MD coat and pretend you're working in the clinic. And so we have these two different patients who arrive in the clinic with these very drastically different symptoms. And so in panel A, you have kind of this skin infection that could be cellulitis or necrotizing fasciitis. In panel B, the patient basically presents with a fever. They have a cough and a chest pain that's been going on for a little while. And the X-ray shows that in one case, this patient has pneumonia. And so you have to think about, what do these patients have in common? Because this is the same infectious agent causing these symptoms. And the diagnosis, you may be in an area where you don't have access to very precise diagnostics. And the time it may take to do the diagnosis is going to be time that maybe this patient with the pneumonia doesn't have. And so in this particular case, a lot of times this might be characterized as like an Acinetobacter infection. And in the case of panel B, this might be diagnosed as TB when the patient comes into the hospital and they're trying to figure out what empiric antibiotics to prescribe. And so these empiric antibiotics are, well, we don't know what you have yet, but we're just going to put you on these antibiotics until we figure out what you have. And so in regions of the world where you may not have access to very precise diagnostics, you're going to take a look at where are these people, what do these people have in common? And so in this particular case, one of these patients might be an agricultural worker. And then 3,000 miles away, you might have a home gardener. These people might be different ages, different sexes, may have no comorbidities, may have different ages, may have some other comorbidities that underlie the disease symptoms that are presenting. And so in this particular case, if you don't know what this patient has for this organism that's infecting these people, time is a big concern. And so you need to rapidly treat it. And so if you treat this patient with drugs for TB, they're going to die in 2 days. And in this case, it might take longer, but the person is going to develop a disseminated infection that's going to go into other parts of their body and be even more difficult to treat. And so the symptoms that I showed you in the previous slides were resulting from an infection with Burkholderia pseudomallei. And so it is the etiological or causative agent of melioidosis. And the example I presented shows you kind of the difficulty in basically diagnosing these diseases, and because it can present with different clinical manifestations. And so in most parts of the world, the diagnostics, it consists of this Ashdown's agar, which is a selective media that basically only allows Burkholderia species to grow and closely related Burkholderia species to grow, and gives it this characteristic red, wrinkly, or rugose colonies. And so this organism has earned this name or moniker as the great mimicker because it resembles all these different infections depending on the route of infection and the host's immune status or other comorbidities that might influence the progression of disease in that host or patient. Another problem with this organism is it has the capacity to basically pump out most of the antibiotics that we have for empiric treatment. It also has the ability to break down beta-lactam antibiotics. And so it didn't acquire these. This is just something that it has to go about its daily business in the environment where it resides. And so it makes it very difficult to treat and results in long courses of antibiotics with limited antibiotics that we can use in our arsenal to treat those infections. This organism is also a facultative intracellular pathogen, which means it can get into cells and it can manipulate those cells to basically reside in those cells. And some antibiotics can't get into these eukaryotic cells, and the immune system can't kill the bacteria once they're in those cells. And so that makes it a very problematic pathogen from a treatment standpoint, and then also for the body to clear the infection over time. And so I mentioned that there are different clinical manifestations that present, and that's based on whether or not the bacteria is acquired through inoculations and cuts, inhaled into the lungs, or ingested into the stomach. And so based on these different modes of or routes of infection, you can get different disease symptoms, and the disease can manifest as ulcers. You can also get lesions, and they can develop pneumonia. And if it's not stopped at these stages, it can get into the blood, where it can then basically disseminate throughout the whole body into every organ, and then also cause sepsis, which is the leading reason why it kills people very rapidly. And so this is a sapronotic disease agent. It's happy hanging out in the environment in kind of stagnant pools of water and soil. And it is a problem for people that have occupations that require them to be in these environments where it is. And so I've shown you a picture here of rice paddy workers. And so oftentimes when people are in the environment and they're aerating the soil, or they're fertilizing the soil, that can put them at risk of basically acquiring this infection. There are also predisposing factors. So anyone that is immunocompromised through immunosuppressive therapy or other infections, or people that have diabetes or defects in lung function, or people that have renal disease, or people who are alcoholics, do have a comorbidity that predisposes them and enhances the ability of this bacteria to infect them. Another important aspect of this is we do see disease outbreaks. They're linked to typhoons and hurricanes and the rainy season in areas where it's endemic. And so this cues you into this idea that aerosolization is a very key component of whether or not people who are healthy acquire this infection. It's also been brought in on consumer goods. And so this was a subject of a Walmart recall a couple of years ago, where there was this aromatherapy spray that was aerosolizing these bacteria into the lungs of people. And the CDC had people return this to Walmart in a bag inside of a box, which is actually kind of frightening if you think about it. And so you can see here that there's plant material in this aromatherapy. And so probably what happened is that the Burkholderia was associated with that plant material and persisted through the manufacturing process. We also see it come in on tropical fish, medical products, and even bottled water. I don't know why we're importing bottled water, but apparently we do. And so in this case, it resulted in sickening 4 people. 2 of them died. The 2 people that survived survived with neurological damage because it went to their brain. It's inhaling it through the nose and pretty much sends it right to the brain. And so this is a neglected tropical disease. You can find it in the subtropics and tropics. This is a great review about where they predict it to be found. Because it's a neglected tropical disease, we don't know exactly everywhere that it's found, but we can predict where it would be found based on the environment that this organism likes to grow. And so it's predicted to be found in 79 countries around the equator in tropical and subtropical zones. And it can lead to 50% or more mortality in places that don't have access to adequate health care for diagnostics and treatment and identification. And so we think that there's about 165 human cases. About 50% of those people succumb to death. And in this particular case, we also think that's just the tip of the iceberg, because a lot of these places where it's endemic, people who die of sepsis, they may not go in and look to see what the cause was. And so you can add the US to that list. So there's probably 80 countries where it's now endemic. CDC researchers investigating 2 cases of people who have never traveled more than 30 miles from their home in Mississippi found that they could find Burglaria pseudomallei in the soil and water around their homes in the Gulf region. And so, using this kind of heat map of where it could be predicted, we know it's here, but it could be in Texas and could be in Florida and other parts of the Gulf. So now it's nationally reportable. And so this is the organism that actually kicked off the whole field of biofilm research. And so in the 1990s, Bill Costerton's group was studying an organism called Pseudomonas pseudomallei for its ability to resist or tolerate antibiotics when it was growing as a biofilm. And so Pseudomonas pseudomallei was renamed to Burglaria pseudomallei. And so I have to remind my biofilm colleagues that this is the original biofilm forming organism that kicked off the whole field of biofilm research. And so in this particular case, Burglaria pseudomallei forms very robust biofilms. And so this is a pellicle biofilm where you have biofilm growing at this air-liquid interface, which will play a role in later in the talk. And so because it provides an extreme tolerance to antibiotics when growing as this structured community, we often see that you can have relapse in patients, and that's been correlated with the ability to form biofilms. After they've been treated with antibiotics, they might show back up in the clinic with symptoms. We also know that biofilms contribute to survival in the environment. Typically, if you're a single-celled bacteria swimming around, there's going to be a bigger fish that's going to come and eat you, because then you're a food source. But when you're growing as this microbial community, you've constructed a safe haven to protect you from all sorts of environmental stresses, bacterial predation, and even in the human host, protection from the immune system. And so I like to show this biofilm life cycle because it'll also play into the rest of the talk, where we have these single planktonic cells, these individual cells. They can either bind to a surface or bind to each other and start secreting these extracellular polymeric substances, which kind of bind them together and allow them to start to form these structured communities. And so once they form these structured communities, they can persist until the environment becomes unfavorable, or some other signal lets them or triggers them to disperse from the biofilm. And they're happy growing in this happy community, but if conditions change, they can disperse or they can break off as aggregates. And in the case of some of these sapronitic disease agents, this is where they can become a problem, is when they're dispersing. And so this polymeric substance, this EPS, this is kind of the core of what protects them from environmental stresses, from desiccation, from disinfectants, from antibiotics. And that's composed of all sorts of things. These sugar polymers that make it kind of slimy, proteins, lipids, and DNA are components of this matrix material that they use to structure their community. And so where this is important is we have these bacteria growing as biofilms in the environment. And at some point, there's something that stimulates them to basically leave the biofilm, where they then become a hazard in this particular case, to human health and animal health, where they can either get into cuts or they can be inhaled. And so in my laboratory, we're interested in understanding these signaling systems and how the bacteria respond to the environmental signals and the conditions that they are encountering and that are changing in their environment that basically trigger them to be dispersed into the environment. And so we've given it the term the great adapter because it has this really amazing ability to sense and respond. And so one of the ways that it does that is through these sensor histidine kinases that are basically protruding out of the membrane that allow them to sense various signals or changes in that membrane to basically alter gene expression through a signal cascade where you have a phosphorylation event, which then results in either changes in transcription or changes in enzymatic activity. And so Burkholderia pseudomallei has 45 of these that are basically sticking out of its membrane, trying to sense what's going on around it. And it can use these primary signals to drive a secondary signaling system, which is basically run by this molecule called cyclic di-GMP. And so if you think back to that first slide where I showed kind of the DNA, cyclic di-GMP is just a smaller packet of information that basically accumulates in the cells to basically drive responses that dictate whether or not the cells decide to form a biofilm or they decide to swim away as individual cells. And so the concentration of this molecule in the cell can dictate this behavior. And the concentration of the molecule of the cell is basically altered by what they're sensing on the cell surface and that signal relay system to drive either making or breaking that cyclic di-GMP molecule. And so I'm interested, and my lab's interested in how bacteria sense and response. And so we take lots of different approaches to evaluate how genes are regulated, genes expressed, and even how proteins are responding to these changes in environmental conditions. And we use a variety of techniques. One of the things that we like to do is look at gene expression under a variety of physiological conditions. Can we change the temperature? Can we put them into a different host? Can we change something in the growth media? Do they differ in biofilms as opposed to planktonic cells? And so one of the workhorses of our lab has been the use of this sequence-defined library of mutants that we built when we first arrived here at CSU. And so we made this library where we could put a transposon insertion in every non-essential gene that the bacteria need or that bacteria have. And so we made this nice arrayed library where we could just go pick our favorite gene or screen the whole library for various phenotypes to understand the specific genes that are important for how this bacteria senses and responds to environmental changes to control behaviors like biofilm formation. And so this was headed up by Grace Borlee before she went off to start her own lab. And what she did was she screened 8,729 mutants, whittled that down to 37 mutants that could no longer form this biofilm at the air-liquid interface. And so from these 37 mutants that no longer formed this pellicle biofilm, we could see that there were different subsets of genes. And so there were genes involved in polysaccharide biosynthesis. And so that was important for that pellicle. And then there's also regulatory genes and metabolic genes, genes involved in swimming. And so if you're going to form a biofilm with that air-liquid interface, you need to be able to swim up and sense that air-liquid interface and start building your biofilm there. And one of the ways that you do that is by attaching to the sides of the surface using Fimbrier. And so in this particular case, there was this exopolysaccharide biofilm gene cluster that we then disrupted. So we deleted it. And so we could do what is called a clean deletion analysis. And we could recapitulate these transposon mutants in a strain that didn't have the other machinery that comes along with making a transposon mutant. And that resulted in disruption of pellicle biofilm formation and alteration of colony morphology that's associated with the secretion of these exopolysaccharides. And so one of the keys to this is that this exopolysaccharide is a hydrophobic polysaccharide. So it allows the bacteria to swim or to basically as like a raft to be able to sit at that air-liquid interface on top of the water. And so that becomes important for when you're thinking about how do things from the water get into the air. First of all, you have to be in a good location for that. And so another thing that we saw in this was there was a hybrid sensor histidine kinase. And so in this particular case, this is a more complex version of that histidine kinase. And we didn't really know what it would respond to. But we started to look at kind of like guilt by association. So if you look at kind of the genome, you can see who's around this particular gene and see what that might interact with and how that might be important. And in this particular case, it was near this diguanylate cyclase 2523. And so one of the things we didn't pick up in this screen was a lot of the genes involved in the secondary signaling or the cyclic di-GMP signaling. And we know that cyclic di-GMP is kind of this nearly universal secondary messenger in gram-negative bacteria that controls this transition. And at the molecular level, we have these diguanylate cyclases that take two molecules of GTP and turn it into cyclic di-GMP. And that results in elevated cellular levels of cyclic di-GMP and a corresponding increase in the exopolysaccharides and adhesins that are important for this biofilm community. Whereas conversely, you can deplete cellular levels of cyclic di-GMP in the cell. And in this particular case, that results in this motile phenotype and cells existing as planktonic. We knew we had 23 ORFs by looking at the genome, doing bioinformatics, that could either make or break or bind cyclic di-GMP. And so we started off by just a simple experiment. Is swimming in biofilm formation altered by cyclic di-GMP? So we could delete one of the phosphodiesterase that we could see, and that would reduce swimming by 50%. And we could basically complement that back because we're good molecular biologists. And we could also look and see what the actual active site is by modifying amino acids in the active site to show that it was a phosphodiesterase that degrades cyclic di-GMP. We could also conditionally express this phosphodiesterase to basically shift the balance from biofilm formation to planktonic cells in our select agent excluded strain. And so what this told us is that these phenotypes are dependent on the levels of cyclic di-GMP. So we then went and pulled all of those 23 genes, and we wanted to figure out whether or not temperature was important. And so I didn't mention this on the other slide when I should have, but temperature and some of the things in the environment are what we're interested in understanding and how that controls biofilm formation. And so we pulled all of those out, and we only got one that really had a strong phenotype at 30 degrees. And it was this 2523 that I mentioned earlier, which is a diguanylate cyclase. And so it gave us this expected phenotype at 30 degrees Celsius, where if we knock it out, the bacteria form less biofilm because they can't make the cyclic di-GMP. What was really kind of paradoxical to us is that if we grew the same panel of mutants at 37 degrees, we ended up with elevated expression of biofilm formation in this mutant. And so what that indicated to us is that, and we could recapitulate that in clean mutants where we went in and deleted them, and that phenotype tracked with concentrations of cellular levels of cyclic di-GMP in the cell. But what that indicated to us was that this was part of a basically a mechanism that the bacteria were using to make biofilms at 30 degrees, but also act as a brake at 37. And that's kind of paradoxical because here you're seeing this elevated biofilm formation. That's because this is absent. It's not there to basically interact with something else to shut it down. And so the other thing that we were interested in is whether or not Burkholderia pseudomallei could respond to nitrate. And so I had this rotation student who was interested in bacteria and all sorts of other organisms. And so having worked with lots of clinical strains, we would always use sodium nitrate in our molecular biology approaches just because it enhanced our ability to manipulate those organisms, but we didn't know why. And so I postulated that that adding nitrate to these soils was shifting the balance from biofilms to planktonic cells. And what Mike did during his rotation was he basically added different levels, and we saw a corresponding response. As you increase the nitrate levels, you see decreased biofilm formation. And so Mike went into a lot of other cool labs, but the thing with Burkholderia pseudomallei is people that work on it seem to become enthralled with it, and he came back. And I had him basically compare the genes for nitrate assimilation and nitrate reductase and nitrate sensing from Pseudomonas aeruginosa, which is the model organism that's very well worked out. And what we decided to do, and what he eventually did, was he looked at this sensory system, this histidine kinase encoded by NAR-X and NAR-L. And when we don't have sodium nitrate, the wild type forms biofilms. The NAR-X and NAR-L mutants form biofilms. But however, if you add the sodium nitrate, wild type basically doesn't form a biofilm, but the NAR-X and the NAR-L continue to form biofilms. So they've lost the ability to respond to this nitrate. And the way that they do this is through this sensor histidine kinase that is sensing some component or breakdown product of nitrate, probably nitric oxide, to trigger the expression of this phosphodiesterase, which then depletes and chews up cellular levels of cyclic di-GMP. So we can measure that through quantitative PCR and concentrations of cyclic di-GMP in the cell. And so this allowed us, and lots of other work that I'm not showing you, to start to develop a model of how these biofilms are formed. What are the blueprints? What is the components that go in to this extracellular polymeric matrix? And we know that there's a lot of polysaccharides that are differentially regulated. There's fimbriae that are important for attachment. But what was important to us is that we could show that nitrate was somehow interacting with this histidine kinase to basically shut down biofilm formation and trigger dispersal. We're also able to kind of get an idea as how they're sensing temperature through this study of this diguanylate cyclase that at 30 degrees contributed to biofilm formation, but at 37 degrees would inhibit biofilm formation, probably in the context of this other histidine kinase, which we yet haven't been able to link the two together, but at 30 degrees, that one does contribute to biofilm formation. And so what's important about all of this for One Health is this dispersal and the response to temperature and to nitrate. And so this led us to more questions about how is Burkholderia pseudomallei dispersed? What contributes to its survival during transit in the air? And so how does it get into the air? How does it survive in the air? And so what you can see here from this map is this is the areas where we know that Burkholderia pseudomallei is. And so this is a consensus map. It's a heat map. So red means there's complete presence, whereas orange means there's good presence, whereas yellow and green might be poor or no identification. And what you can see overlaid with this aerosol optical depth model from NASA is that a lot of these regions where you see this kind of airflow and transit, in this particular case, we have dust in orange, we have organic and black carbon in green. You can also see kind of the tropical storms and the typhoons that are pushing through these areas where you have it endemic. And so to me, that suggested that there is a very significant component to this organism being emitted into the atmosphere as bioaerosols. And we know that after heavy rains and typhoon events, that's when you see outbreaks in these areas where it's endemic. And so that led us to try to figure out, well, how can we study survival in the air? How does it get into the air? And this is kind of plug for melioidosis as a poster child for One Health aerobiology. There's a good review here if any of you are interested, just because it is this organism that basically affects all sorts of different animals. And it does affect people that have various comorbidities, people that are in the environment being exposed to it as agricultural workers. And it is going to be one of these things that basically increases its incidence and where it is in the environment and expands its territory based on this idea that we have changing weather patterns leading to more extreme weather. And so one of the things that we did is we wanted to model this in the laboratory. And so a bunch of us put in for this Catalyst for Innovation project. So this is a SIP project that was sponsored by our OVPR. And this was led by Angela Bosco-Loth and Jan Leach and Sonia Krendenweiss. And the idea was to bring different people from different perspectives to basically try to understand some important biological principles that we could model in environmentally regulated growth chambers that were built by our engineering students as their senior engineering projects. And so Jan Leach was a expert on rice, and Sonia Krendenweiss is a engineer, and Angela Bosco-Loth is a person who's really interested in modeling disease in animals. And so we use this approach to start to get at the idea of how are these bacteria sensing and responding to the environment. And we built this enclosed ecosystem where we could study the effects of this bacteria and dispersal of this bacteria in this controlled environment. And so in this particular case, we use the surrogate organism Burkholderia thailandensis, which is what we would call a safe relative to work with as compared to Burkholderia pseudomallii. It survives in the same environment. They compete with each other, except thailandensis is missing some of the key virulence factors that pseudomallii is. And so this would be part of this Burkholderia pseudomallii complex, but yet doesn't cause the severe disease and the sepsis that you would see in humans and animals. And so this turned out to be a bit of a Pandora's box for us, just because we put the Burkholderia thailandensis in, we could track it using bioluminescence markers, we could track it using a selected media, that Ashdowns media that I showed you previously, and we could also just sequence to determine if it was there in the population. And what happened is it rapidly disappeared and became undetectable in our ecosystem. And so this was a problem for us because we were trying to understand, how this organism disperses in the environment, but yet it's just disappeared. And so what we found through subsequent culturing is we had this unknown bacteria that was basically interacting with our Burkholderia thailandensis, or our safe surrogate. And we sequenced it, and lo and behold, it was pretty closely related to Burkholderia, but yet not. Yet it is its own distinct genus and species. And so we published that sequence, and then we wanted to figure out, well, how is this Pandorea numinusa interacting with Burkholderia thailandensis in this rhizosphere community? So in the context of those roots of the rice that I showed you. And one of the things that we found is that the thailandensis itself, when we add it to the roots, basically disappears rapidly. And so that correlates to what I told you earlier over time. And if we add Pandorea numinusa to that, it disappears even more rapidly. And so the Pandorea numinusa was accelerating decolonization. However, if we basically sterilized the rice seeds prior to germinating them, we could then get a stabilized community of the Burkholderia thailandensis when we added it. However, if we added the Pandorea numinusa and the Burkholderia thailandensis at the same time, the Burkholderia thailandensis would just disappear. And so that suggested to us that we had isolated an organism that can compete with the Burkholderia thailandensis. And, that was at the end of our project. But what was great about that project is it brought together these people from diverse disciplines. And we had like monthly meetings and seminars, and having that ability to interact with each other allowed us to go after some bigger questions. And so that group of people then expanded and brought in more people of more disciplines and diversified that team to go after this NSF grant in a Biology Integration Institute, which we named our institute Biology Integration Institute Regional One Health Aerobiome Discovery Network, or just broaden. And so with the idea that we were going to study what's in the air, what it's doing in the air, and how that affects the ecosystems, whether or not it affects human and animal health, how do these bacteria get there, how do these microbes get there, and how do they survive once they're there. And so one of the great things about this, having this multidisciplinary team, it allowed us to focus on some different aspects of what we thought was lacking in aerobiology. And so aerobiology has been around for a long time, but the standards for air sampling and analysis can be a little bit diverse, we'll say. And so what we wanted to do was contribute to establishing some international standards for air sampling and analysis of the organisms that are there. We also wanted to develop some modeling framework for predicting how the composition of the communities that are in the air change and what stimulates those changes. Is it during night? Is it during the day? And then we also wanted to be able to take the information that we've garnered here and bring that back to the lab to understand how these bacteria are getting into the air and how they're surviving once they're there. And this allowed us to team up with other people that have other resources, so air sampling resources, chambers to actually conduct these studies under controlled conditions, laboratories to actually do kind of these mechanistic, genetic type studies, sequencing, high throughput sequencing. And so in the case of goal 3 here, where we want to look at the mechanisms of emission and survival, there's lots of different projects and different labs surrounded on that. What I'm going to talk about today is this idea of how rhamnolipids can contribute to aerosolization and bioaerosol generation. And so this focuses on kind of these first step in these 3 stages of the bioaerosol life cycle, with the idea that the bacteria have to either get off of the terrestrial landscape in some fashion to actually get into the air so that they can be transported and deposited and then brought back down to the ground to start this cycle again. So we're going to be focused on emission. And so there are lots of different drivers of bioaerosol generation, or how these bacteria can get into the air. And so the ones that I'm going to focus on are kind of this idea that, you can have raindrops that basically result in impact and splash and eject droplets and whatever particles they pick up into the atmosphere. And so that can happen on surfaces, plant surfaces, soil, and even water films. And so I've been showing you a couple of different water films throughout this talk. So this pellicle biofilm could be considered a water film. And sometimes in nature, you can see these organisms that are making, in this case, an iridescent sheen. That's not oil, that's actually microbes. And so how does something like this or this get ejected into the air? And a lot of it is thought to be through either like rainfall and splash or the bubbles bursting. And so here's just an example of like these waves crashing, where that's basically causing emission of sea spray aerosol into the air. bubbles rupturing. And so in this particular case, these surface properties of these biofilms associated either with a dry surface or even a wet surface or a water film are important. And so we were interested, can surfactants produced by the microbes contribute to this idea of emission? And so surfactants are these hydrophobic and hydrophilic molecules that basically lower the surface tension of these microbes in contact with surfaces. And in the case of bubbles, these surfactants will promote bubble persistence. So you'll get these bubbles that last longer. So this is one where they've added surfactant and one that doesn't have surfactant. These bubbles maintain the bubble surface longer before they break up. And in this case, they're releasing more particles as droplets. And so these surfactants are known to alter kind of bubble formation and emission when bubbles rupture. And so we wanted to test the role of rhamnolipids. And so we know that in the bacteria that we work with in my lab, these Pseudomonas and Burkholderia species, they make a couple different rhamnolipid biosurfactants. We wanted to know if it alters the emission dynamics during bioaerosol generation. Does it alter aggregate size and droplet size? And so the goal here was to test mutant strains that cannot produce rhamnolipids in these sapronitic disease agents. And so I talked about how we've used Burkholderia thailandensis as kind of this safe neighbor or surrogate for Burkholderia pseudomallei. But we can also use Pseudomonas aeruginosa, which is also a sapronitic disease agent that we can use in the laboratory as a model organism. And so this is a ubiquitous sapronitic disease agent. It's found in diverse environments. You're coming into contact with it on a daily basis. And it usually doesn't alter, it doesn't harm you unless it gets into the wrong place at the wrong time or you become immunocompromised. And so you can find it in rivers, you can find it in soil, you can find it breaking down petroleum products, you can find it associated with insects and associated with your dogs. And it's pretty similar in its lifestyle to the Burkholderia species, which also can do all of these things, where it could set up as an infection, or it can be, something just cycling nutrients in the environment. And so in Pseudomonas aeruginosa, we know that rhamnolipids can be important for enabling surface motility, where they're breaking the surface tension. So this is a wild type strain of Pseudomonas aeruginosa. It's forming these tendricles as it swarms out. You can knock out rhamnolipid production. It doesn't have that ability to swarm across surfaces because it can't break the surface tension under those conditions. It can also allow for emulsification and uptake of hydrocarbons, so petroleum products. We know it's a virulence factor, but what's interesting from my standpoint is that these rhamnolipids act as a surfactant to create kind of these void spaces in these structured communities to allow them to organize these channels, which are important for biofilms, to bring in nutrients and oxygen into those biofilm communities. We also know that when you compare wild type Pseudomonas aeruginosa biofilm to one where you can induce rhamnolipid production, that initiates dispersal from the biofilm. And if you're looking under the microscope, these little planktonic cells look like an angry beehive, where they're basically getting ready to disperse, and they're swimming around inside of that biofilm community that is deteriorating. So we wanted to know, does Pseudomonas aeruginosa rhamnolipids contribute to bioaerosol generation? And so this was a team approach with our colleagues, led by Mark Hernandez at CU Boulder and at their shared instrumentation facility where they can do aerobiology in aerosol chambers. And this efforts were led by Sam Golan, working with Marina here at CSU to develop this idea or this technology to mimic bubble bursting in a chamber to understand whether or not our specific mutants could survive. And so this is just a picture of why you need microbiologists need engineers as friends, because there's all these different pieces of equipment that are required for generating these bioaerosols, for detecting and capturing these cells so that we can determine whether or not they're viable? And then also, can we determine what size they are? And, are we applying constant rates between treatments? And so what Sam found was that when he takes his wild type strain of Pseudomonas and then takes a mutant of that strain and then nebulizes it, he sees a significant reduction or drastic difference in the number of recoverable CFU between the strain that makes rhamnolipid and the strain that's deficient in rhamnolipid production. And so in this particular case, probably about a 10% reduction in the CFUs. And since he has an annoying PI, I'm like, well, we need another way to look at that other than just this rhamnolipid mutant. And so in this particular case, I was kind of asking, well, how do these surfactants get out? And it's pretty much unknown. However, Sam was able to show me that these R&D efflux pumps, which I mentioned earlier, can be co-located with the rhamnolipid biosynthesis genes in Burkholderia pseudomallei. And so that led us to the hypothesis that these rhamnolipids are getting out through these efflux pumps. And so when Sam looked at the efflux pump mutant, he found that it doesn't produce or doesn't secrete surfactant. And when he took these mutants to the chamber, he also found that there is a reduction in the recoverable CFUs when you compare a wild type strain with a efflux pump mutant strain in Pseudomonas aeruginosa. And so just to kind of tie this all together, one of the things that I've shown you today is that you can understand these environmental sensing pathways in the bacteria to understand how it shifts their behavior in response to important signals like temperature as an organism infects a warmer human host, or nitrate where they might be in the environment and stimulated to leave that environment to basically then disperse and infect hosts. And so one of the key takeaways I'd like you to all have is that integration of diverse expertise and investigation teams and the resources that they can bring to those collaborations allow us to answer questions that we otherwise couldn't answer as individual labs. And so with that, I'd like to thank our broadened team, thank our funding from NSF, thank the support that we get from One Health Institute here to actually run these programs, run this program. I'd also like to thank the current Borlee Lab members that do research, former Borlee Lab members, a lot of the collaborators that I have in the Burkholderia field around the world, and then some of the CSU collaborators that are integral to this research. And so with that, I'll take any questions. And this is our support that's driven our lab for 14 years. So.
And for questions, both online, which will monitor the chat, and in the room, and I have to go teach, so Jessica will lead that part.
Could you comment about whether the disease is increasing in frequency? So
there are reports of it increasing in frequency based on these climactic events. And so with more typhoons and more drastic rainy seasons, you are seeing the incidence increase in those areas where it's endemic. And I think now that so now in the US, they have to look for it and report it. So that will ultimately factor into an increase in incidence reporting here.
You were talking about how the BP has the antimicrobial efflux pumps.
Mm-hmm. So
is that older than antimicrobial resistance? Like that, why is that part of its evolutionary history?
Yeah, so there's recent papers where they're going into these soil samples from 50,000 years that haven't seen the light of day, and they find these organisms with efflux pumps. And so these efflux pumps are just a natural part of their metabolism. So that's why we think, this is why the rhamnolipids are getting out. But if you challenge the bacteria with an antimicrobial, it will adapt over time to basically pump that antimicrobial out. So these efflux pumps are there to pump out things out of the cell that the cell makes, whether it's something important like a rhamnolipid or maybe even a waste product or some molecule that's derived through secondary metabolism.
Thank you, first of all, for the presentation. When we talk about Borrelia or other bacteria that can parasitize on animals, but also live in the environment, it's always main questions, what's going on when during this transition from parasitic phase to saprophytic phase? And two main points, or either it's selective pressure of some variant strains or switch of enzyme systems. But first, the selective mechanism is more attractive because you may have some markers to track where you've got, let's say, in the air, what source from. When you're talking about like wild type, it's not clear. You just pick this and you might some experimental manipulation. But what is original? What is difference from animals or from the rules of play?
Yeah, so I can speak to that because, and it's a great point that you bring up. And so in the case of like Pseudomonas aeruginosa, which is a sapronotic disease agent, patients with CF will basically acquire just random environmental isolates. In the case with Burkholderia pseudomallei, it's just a random environmental isolate, but it has this ability to sense and respond that then allows it to understand, well, I'm in this environment, I need to make this to get out. So if you're a bacteria trapped in a CF patient's lungs, maybe that's not where you want to be, but you have to get out somehow. And so one of the ways that they do that is by expressing a lot of these virulence factors, which have probably evolved for them to basically compete with other organisms in the environment. And so in the case of like Pseudomonas aeruginosa, it doesn't play nice with other organisms in the environment. It has a whole arsenal of weapons that it uses to kill fungi and bacteria that are basically in its space. And so unfortunately for us, evolution of that arsenal also contributes to its ability to infect us.
About Borrelia species, they can coexist together, but they can colonize together.
Yeah, so the Borrelia thailandensis and the Pseudomallei can coexist, but they do have ways of discriminating between each other and basically deciding whether or not they like each other or don't like each other. And in those cases, they have these secretion systems that will poke holes in each other, like dueling swords. But they basically evolve those based on whether or not they can coexist or whether or not they're competing with each other.
A question. Did I hear you correctly that adding the nitrate to the soil caused them to leave the soil? Or
Yeah, and so it causes them to disperse from the biofilm where they're happy.
Okay.
And so I can go into the other aspects of what nitrate do, but nitrate will turn on a whole bunch of virulence factors too. So not only does they leave the biofilm, but then they turn on all these things that actually can cause damage to eukaryotic cells.
Okay, because then I was curious with nitrate that will, with obviously with runoff, we're talking soil, that contributes to algae bloom. Will that bacteria then flow through that cycle into that algae bloom and then be dispersed through the algae bloom?
So they might then set up a biofilm, and then if you have kind of a weather event, or maybe goes over a waterfall, or anything that is disrupting that biofilm to eject it into the atmosphere, is where it then becomes a problem. And so if they're in a biofilm, that's going to resist this propensity to be ejected. But if they're basically triggered to go into like the single cell planktonic state, then that makes them more efficiently aerosolized. Thanks for all the questions.
Thank you so much. We really appreciate it.
Weren't any online, so we're good.